I'm Kaelyn Fogelman, and I'm a PhD candidate here in the School of Fisheries, Aquaculture, and Aquatic Sciences, and I am also the lab manager of Dr. Jim Stoeckel's Crustacean and Molluscan Ecology lab.
I'm Kayla Boyd. I'm a PhD student in Dr. Stoeckel's lab, and I'm from Wisconsin.
So my research specifically focuses on mussel feeding ecology. As we know, freshwater unionid mussels are one of the most imperiled group of organisms we have in North America, with the exception possibly of snails, but we don't have a good idea of what these animals eat in their natural systems. I try to find out what the mussels are eating. So I sample, food from the water column because they filter, and then I also sample food that's on the bottom of the lake or the river so that I can try to see, are they getting the water and the food from where they're sitting and they can access maybe with their foot, or are they filtering all of their food from the water column? I do the same thing, in my rivers as I do in my lakes, and I have, endangered species, and we need to know what these animals are eating so that we can successfully grow them in captivity, we can propagate them in captivity, and we can re-release them into the wild so that we can bolster these populations that we, are declining over time. So the work that I do is in rivers in Texas, so these are lotic systems or flowing systems, and then I also do work in a lake in Alabama. So this is a lentic system, or a pond, a reservoir, something that's not flowing like a river. So for these individuals, what I found is in my rivers in Texas, they are primarily getting their carbon source. That's a way that we detect what their primary food sources are, is we look at their carbon and nitrogen and how it's assimilated in the animal from the environment. So in our Texas rivers, they are getting their primary carbon source from leaf litter that's coming from outside of the system, and in my Alabama lake, all three of my species are getting their primary carbon source from the sediment that they're living in down on the bottom. And when we have these animals in captivity, this is important now because we need to give them some type of substrate to feed on and access as well, because we know they're getting a significant amount of their food from this substrate that they're living in.
I study biocontaminants, specifically biocontaminants that you can find in aquatic systems. How these biocontaminants work their way into these aquatic systems is through things like wastewater discharge, or the discharge from manufacturing and industry, or water runoff from rains, and they end up in our rivers and our lakes and our ocean. So a lot of my work focuses on bioenergetics, which is the idea that animals, when they take in their food, that is the energy that they use for things like growth, things like reproduction. They use a little bit for metabolism. Stressors can decrease the amount of energy that those individuals now have to use for growth and reproduction. Pollutants, when they enter an animal's body, sometimes they accumulate within the tissue, something we call bioaccumulation. And animals, particularly oysters, are very good at taking these chemicals out of their tissues and removing it. But that act of doing that is incredibly expensive, or can be depending on the contaminant. So what I look at in particular is how expensive this is for oysters. And so within that framework, I can expose animals to these contaminants. I can expose oysters, have them bioaccumulate these chemicals for a certain amount of days, and then I can get their resting metabolic rate, which is the amount of energy that they need to use for general cellular growth and maintenance. And then we can determine, if we were to do an assay that would show how much energy the animal is capable of using, we can then determine if these chemicals are reducing the amount of energy that that animal now has to grow, reproduce, and things like that. The biocontaminant work that I do for the oysters is PFAS in particular, and all of the preliminary results so far with my work has been incredibly exciting. We've been seeing that the bioenergetic effects on the oysters between those that are exposed to these contaminants and those that are not have been non-existent. So essentially, whether they are exposed or not, the act of depurating is not energetically expensive to those individuals, which is a great sign for animals that are out on the reefs and also animals that are on the farms.
So the work that we do specifically in the Crustacean and Molluscan Ecology Lab is applicable specifically to Alabama, because in Alabama we have the highest biodiversity of freshwater animals in North America, and oftentimes for some of this taxa that we work with, we have more biodiversity here than anywhere else in the world. So for example, Alabama has more crayfish and mussel species than any other state in the country or the rest of North America. So this is important also because 50% of crayfish are imperiled and 70% of mussels are imperiled. So the work that we do here ties back into this biodiversity because we have more aquatic animals than anywhere else, and it's our duty to conserve them.
Alabama also has a very large shrimp and oyster industry that's growing right now on the coast, particularly oysters. A lot of the work that we do is also not only applied to native systems, but can also be applied to agriculture systems. Since the reefs and the farms are so close to each other, a lot of the results can transfer between the two groups, between agriculture and conservation.